Good evening, colleagues. Our apologies for starting late. There's still lots of people entering, so I'm just going to give a minute or so, and then we will get going. Okay, right. Welcome, everyone. Welcome to another session of the CHM CPD program. This evening, I have the pleasure to introduce Dr. Michelle Bisschoff. Dr. Michelle Bisschoff is a postdoctoral researcher at the Center for Human Metabolomics, Northwest University, Potchefstroom. She holds a BSc degree in Environmental and Biological Sciences, summa cum laude in 2013, and a BSc Honours in Biochemistry, summa cum laude 2014, an MSc in Biochemistry, cum laude 2017, and a PhD in Biochemistry, all from the Northwest University. Dr. Bisschoff has 9 years of practical experience in bioenergetics, molecular biology, enzymology, and cell cultures. Research interests focus on inherited errors of metabolism, particularly mitochondrial and metabolic disorders affecting young individuals. She is currently involved in the International Center for Genomic Medicine and Neuromuscular Diseases, a transcontinental research initiative focused on gene discovery and disease mechanisms of neuromuscular disorders like multiple acyl-CoA dehydrogenase deficiency, which is also the topic of her talk this evening. Dr. Bisschoff is dedicated to improving the health and quality of life of individuals affected by inborn errors of metabolism through her research. Michelle, welcome, and we look forward to your presentation this evening.
Thank you so much, Prof, and thank you for the opportunity to present my work. So tonight I'm going to talk a little bit about my PhD thesis, which is entitled The Biochemical and Molecular Genetic Characterization of MADD in a Southern African Cohort. So to start us off, I'll just give a brief outline of the presentation. So first we'll start with an introduction to MADD. This will be followed by the study rationale and aims. Hereafter we'll have a look at the study design and results. And finally, we'll come to some conclusions. So multiple acyl-CoA dehydrogenase deficiency, or MADD, is a rare inborn error of metabolism. It was formerly called glutaric aciduria type 2. And the first case was described over 40 years ago. So birth prevalence of this disorder is 1 in 200,000 live births. However, there is a lot of variation reported in different countries and with ethnicity as well. Genetically, MADD may be caused by variants in 3 genes. These are ETFA, ETFB, and ETFDH. ETFA and ETFB code for the alpha and beta subunits of the electron transfer flavin protein, and ETFDH codes for ETF ubiquinone oxidoreductase. Of these, the variants in the ETFDH gene are most commonly encountered, with more than 347 recorded to date. So to explain a little bit how these genes and the proteins that they code for work together, we have to look at the ETF-ETFQO system. So the purpose of the system is to reoxidize reduced flavin adenine dinucleotide, or FADH2, to its oxidized form, FAD, for use by a variety of different enzymes. So the ETF-ETFQO system couples to various other enzymes and links them to the mitochondrial oxidative phosphorylation system. And in this process, FADH2, which is produced by these the variety of other metabolic pathways that are coupled to this, apologies, is then oxidized firstly by reducing ETF, and then ETF transfers electrons to ETFQO, which is then reduced, and the reduced ETFQO then finally transfers the electrons to coenzyme Q10, which then transfers them to the mitochondrial oxidative phosphorylation system. They then travel through the system and finally generate energy in the form of ATP. So to really understand how far the system reaches, we have to look at all the enzymes that are involved that require FADH2, and these enzymes are known as flavoproteins. The majority of these flavoproteins are involved in mitochondrial metabolism. They are also in other sections of the cell. But in the mitochondrion specifically, they partake in choline metabolism, fatty acid catabolism, branched chain amino acid catabolism, and amino acid catabolism. And all the enzymes shown here in the boxes, these enzymes are all involved and require oxidized FAD to function. So if oxidized FAD is not available to these enzymes, the individuals will develop toxic metabolic buildup in these metabolic pathways. So I'm not going to go into detail about this, but these are just some of the metabolites that can be increased in individuals with MADD. Right, so MADD can present as one of three phenotypes. Types 1 and 2 are neonatal phenotypes. They are classified as severe to fatal, and these have multi-system involvement. So the symptoms can include cardiomyopathy, coma, hypotonia, lethargy, liver dysfunction, rhabdomyolysis, skeletal myopathy, muscle pain, and muscle weakness. These individuals also have metabolic aberrations, including hypoglycemia, hyperammonemia, metabolic acidosis, and sometimes ketosis as well. Type 3 is a later onset phenotype, which is mostly encountered in children, adolescents, and adults. It has a milder presentation, and it presents, or it can present, with all of the symptoms encountered in type 1 and type 2, as well as a few other more common symptoms. So these symptoms include vomiting, lipid storage myopathy, exercise intolerance, again, muscle weakness, and then the typical metabolic symptoms. However, ketosis is not always present in these cases. One of the ways to distinguish type 1 from type 2, apologies, is that type 1 has congenital abnormalities. Type 2 never presents with this. Right, so MADD can be diagnosed by broad spectrum metabolic testing. This is also done in South Africa. Additional enzymatic flux assays can follow this. This is typically done in countries that where MADD is more actively studied. We do not perform this in South Africa. An important factor to consider, however, is to genetically confirm metabolic disorders like MADD. And the reason for this is that MADD typically only presents with symptoms and metabolic derangement during the catabolic state. So in the anabolic state, a patient may appear completely normal if their phenotype is not severe enough. So the ideal sampling time for metabolic testing is then during the catabolic state or metabolic crisis, and for neonates, it'll be before the next feeding. And typically, sampling should always be done following minimal medication intake. Another way that we can gauge the severity of MADD is by following the MADD DS3 disease severity scoring system. Now, this is a recently developed scoring system that was developed in the Netherlands based on retrospective studies. This system is based on the weighted opinions of a panel of expert clinicians that have dealt with this disorder for many years. And the results of this specific scoring system correspond to what is observed in the patients, as well as what is observed in the laboratory. If we look at treatment, MADD is very amenable to treatment. All of them are treatable apart from MADD type 1, which unfortunately has not yet been treated with success, and very severe cases of MADD type 2. But there are cases of MADD type 2 that respond well to treatment. So for MADD types 1 and 2, emergency treatment will include a high-dose glucose infusion with insulin, as well as L-carnitine supplementation if there is any reason to believe that the carnitine supply is low. In terms of MADD type 3, the emergency treatment includes riboflavin. And then standard treatment includes riboflavin for all three of these phenotypes, the strict avoidance of fasting, a high carb diet, a diet that is low in protein and low in fat, and then L-carnitine supplementation in the case of carnitine deficiency. There's also some experimental treatment. This includes coenzyme Q10, which may be given to decrease reactive oxygen species. Reactive oxygen species may occur in these individuals as MADD may lead to mitochondrial damage. Another experimental treatment is 3-hydroxybutyrate, which is a ketone body, and this can be given to individuals and has actually been met with great success, and it acts by just replacing the fuel source. So instead of using fatty acids, for example, the individual then uses ketone bodies more efficiently. Right, so if we look at the rationale behind this study, MADD is very underdiagnosed and poorly studied in Southern Africa. While other countries focus more on newborn screening to pick up disorders like MADD, Southern Africa does not have this option, and diagnoses of MADD, as with other rare metabolic disorders, are mainly made on an elective basis. So this follows the request of a parent or physician's referral. In addition, we do not perform genetic confirmation routinely in our country, and all of this leads to lacking information regarding the phenotype, genotype, and metabolic profile of MADD, as well as the pathogenicity and functional consequences of variants. So previously at our institution, research indicated that 2 ETFDH variants might be quite common in our population. This included the 1448C2T variant and a novel variant, the 1067G2A variant. We also found that this novel variant, or we also hypothesized, that this novel variant may be the result of a potential founder effect. MADD is, we also, based on the anecdotal findings and the small cohort that we had, we hypothesized that MADD might actually be more prevalent in Southern Africa. So to address this, we worked together with the International Center for Genomic Medicine and Neuromuscular Diseases, or the ICGNMD. So the ICGNMD is a UK-headed product. It's based in the UK with collaboration of 3 universities, from one of them being Newcastle University, then the Cambridge University and the University College London. And the focus of this project is to work together with 5 understudied countries, including Southern Africa, with focus on neuromuscular disorders to improve diagnosis hereof, and to develop a better genetic platform to do this as well. So in South Africa, the collaborators of the study included 4 universities and 4 academic state hospitals. So this was the Northwest University, specifically the Potchefstroom campus, then the University of Pretoria in collaboration with the Steve Biko Academic Hospital, the University of Cape Town in collaboration with the Red Cross War Memorial Children's Hospital and Groote Schuur Hospital, and then Stellenbosch University in collaboration with Tygerberg Hospital. And of course, all of these hospitals had at least 1 pediatric or adult neurologist. Right, so to address what we wanted to focus on in this study, then the study had 2 main aims. The first was to characterize the clinical, metabolic, and genetic properties of MADD in a Southern African cohort, as that this has not been done in a large cohort before. And then the second was to evaluate via in vitro analyses the functional properties of pathogenic variants that were identified most frequently in this cohort. So if we have a look at the study design, the first step was to select a cohort, and this included probants of affected individuals and their first-degree relatives. This was done following informed consent and with ethical approval by the 4 Southern African state hospitals, as I've just mentioned. Inclusion criteria included South African citizenship, Southern African citizenship, apologies, and a clinical MADD diagnosis and exclusion criteria was a positive diagnosis of HIV. The reason for this being that HIV and its treatments are frequently associated with neuromuscular symptoms, or they mimic neuromuscular symptoms rather. So from these individuals, samples were then obtained. For genetic analyses, this included saliva or EDTA blood from living individuals and urine from individuals who had been deceased prior to the onset of this study. For metabolic analyses, this included urine that was collected at first presentation and following treatment. For the allele frequency analyses, we used newborn dried blood spots and umbilical cord DNA. And for the functional analyses, we collected skin biopsies from specific cases. Hereafter, thorough clinical evaluations were conducted by adult and pediatric neurologists, and this was done by standardized ICG and MD protocols. For all of the individuals, as well as their family members, genetic analyses were performed. So for all of the affected individuals, whole exome sequencing was performed, and to confirm the mode of inheritance of the specific variants that we detected, trio analysis was done using Sanger sequencing. The variant pathogenicity was then classified according to the ACMG criteria. Right, so if we have a look at the results, we were able to obtain a cohort of 14 participants. Unfortunately, 3 of these participants had passed away before the onset of this disorder. Before the onset of this study. The cohort consisted of 5 males and 9 females, and they came from 12 unrelated families. Self-reported ethnicity included 12 Caucasian participants and 2 participants of mixed ethnicity, and no consanguinity was observed. Following whole exome sequencing, 5 variants were identified in the EGFDH gene. This was as expected. As I've mentioned before, the EGFDH gene is the most commonly affected in MADD. So the first was the 287 duplication variant, which was a novel finding and has only been reported in this cohort. The second and third was the 740G2T and the 976G2C variants. These have only been reported each in a single other case in a Chinese cohort. And then the 4th and the 5th variant were the 2 variants that we identified previously in our own study, which was the 1067G2A variant and the 1448C2T variant. Right, so if we have a look at the variant pathogenicity, if we firstly look at the novel variant, the 287 duplication variant, this variant is encountered in a one of the very early exons of the gene, the 3rd exon, and it also leads to a premature stop codon, which results in a protein which is much smaller than the native protein. All the other variants also lead to amino acid changes, but they occur much later in the protein and they do not lead to truncated forms. They occur at various places in the specific protein, but all of them occur in regions that have functionality. So that indicates that these variants might have a pathogenic role. Then if we look at the ACMG classification, the widely reported 1448C2T variant had a classification of pathogenic, whereas all the other variants had a classification of likely pathogenic. And all of these variants had either one very strong or one strong evidence or ACMG classification evidence with 1 or 2 moderate or supporting categories for evidence. So from this, we could say theoretically that all 5 of the variants we identified were very likely going to have pathogenic results. If we then look at the clinical investigations, it was possible to group the cohort based on disease severity and the clinical symptoms into 3 groups. So the first was the riboflavin unresponsive group. This group consisted of 3 infants, 1 Caucasian male and 2 females of mixed ethnicity. Onset was at birth, and these individuals unfortunately died very shortly after birth. The major systems affected included the liver and the muscle. And in the case of the male infant, congenital abnormalities were also present. Considering this, together with the MADD-DS3 scores that we obtained, the male infant was given a diagnosis of MAD type 1, while the female infants were given a diagnosis of either 1 or 2. And the reason for this being is that congenital anomalies could not be ruled out in these 2 cases, since it could not be established whether or not these individuals had any congenital issues. So the second group included the variably riboflavin responsive group, and this included 8 individuals. In this group, we had 4 males and 4 females, all of them Caucasian, and onset ranged from birth to adolescence. So it was quite wide. All of these individuals responded to treatment apart from 1, and that was participant 7. So if we look at the organ systems involved, it was very heterogeneous, included the muscle, the liver, the central nervous system. And in the case of participant 7, these symptoms unfortunately progressed, and this participant succumbed to her symptoms at the age of 23. However, unfortunately, it was not, we were unable to establish whether this individual had succumbed to symptoms, or let's rather say that this individual had a stroke, and it was not possible to determine whether the stroke was specifically due to MADD or due to a history of this. So if we then look at the third group, this is the riboflavin responsive group. This included 3 females, 3 Caucasian females, and they all had an onset or a later onset, one of them as a child, but the other 2 as adults. Again, the symptoms in this group were very heterogeneous, and the MADD-DS3 scores were a little bit lower apart from participant 5. However, in the case of participant 5, the MADD-DS3 score was influenced by this participant's self-reported severity. She was diagnosed very late in her life following a lot of complications, so she was hospitalized, and this unfortunately increased her MADD-DS3 score. So that's what I mentioned earlier, that this MADD-DS3 score, while it is very helpful, if it's applied to individuals who have been diagnosed later in their lives, it might not always be accurate. So importantly, the riboflavin responsive group was very responsive to treatment, and all 3 of these cases were assigned a phenotype of MAD type 3. So if we have a look at the genetics, if we combine this, we can then see that the 1067G2A variant and the 1448C2T variant was present in one form or another in all of these cases. So these 2 variants really are the 2 most frequently occurring in our population. In the first group, the riboflavin unresponsive group, we can see that only 2 genotypes clustered to this group. That was the homozygous 1067G2A genotype and the 976G2C 1067G2A genotype. Both these variants and these genotypes occur in the ubiquinone binding region of the ETFQO protein, and it is hypothesized that because both of them occur in the same binding domain, that it has an additional or larger pathogenic effect, leading to complications in these participants. If we have a look at the variably riboflavin responsive group, we can see that these individuals presented with heterogeneous, with compounded heterozygous genotypes. So this included the 1067G2A, 1448C2T genotype, then the 287 duplication 1448C2T genotype, and the 740G2T 1448C2T genotype. Of these, the 1067, the one that's written at the top, the 1067 1448 genotype is hypothesized to be the more severe in this specific group. And then if we have a look at the riboflavin responsive group, we can see that all of them presented with the homozygous 1448C2T genotype. Right, so next, after we did the clinical and the genetic analyses, we had a look at the metabolomic analyses of these participants. So to do this, I mentioned earlier that we require urine and plasma samples. So while plasma samples are preferred, urine samples was what we had available for the majority of this cohort, since this cohort was diagnosed over a period of more than 20 years. So back then, the plasma analyses that we had were not comparable to the ones that we have today. So we decided to work with the urine metabolic data that we had. So for the metabolic analyses, we then did or analyzed the organic acids using GCMS, the acyl carnitines using LC-MS-MS, and the amino acids using LC-MS-MS with chromatographic separation. To then have a look at the two variants that I mentioned earlier, the 1067G2A and the 1448C2T variants, to have a look at their allele frequency in Southern Africa, we then performed PCR-RFLP analysis on healthy controls. So these healthy controls is the newborn dried blood spot and umbilical cord DNA samples that I mentioned earlier. And we did this in the 4 largest population groups of Southern Africa, so the African, Caucasian, Indian, and mixed ancestry groups. The samples were an equal number of male and female samples, and they represented both the northern and the southern parts of Southern Africa. We then looked at the haplotype, or did haplotype analyses for these 2 specific variants as well. And for this, we used a SNP array, and we performed this on the participants' DNA that specifically presented with these variants. Right, so in terms of the metabolic investigations, firstly, if we have a look at the riboflavin unresponsive group, again, these results clustered to the 3 groups that we were able to form based on the clinical aspects of this cohort. So if we first have a look at the riboflavin unresponsive group, you can see that the organic acids and amino acids are quite high, the acyl carnitines as well in this group, and that this did not appear to improve considerably following treatment. There was an improved excretion of acyl carnitines in this group, as you can see from the treated, the increase in the treated values that we have here, but unfortunately there was no improvement in organic acids or amino acids, and unfortunately these individuals succumbed to their symptoms very early. If we then have a look at the variably riboflavin responsive group, it looks a little bit better. In the untreated samples, you can see that it's a lot lower. However, in this group, it should be noted that the metabolites that were detected were more heterogeneous. So They still corresponded to what you would expect for a MADD metabolic profile, but there was a lot of variation. Treatment improved the outcome, the metabolic outcome of these participants. And again, specifically, the L-carnitine treatment had a good effect on the acylcarnitines. Then if we have a look at the riboflavin responsive group, you can see that these values are much lower. For some of the individuals here, the values were high, especially participant 5, but as I've mentioned, she decompensated quite a bit prior to her diagnosis, and so her values are not what we would typically expect for an adult with riboflavin responsive MADD to be. Following treatment, these values essentially normalized. They were much better, and these individuals carried on with treatment without any further complications. Right, so if we then have a look at the allele frequencies for the two variants that were encountered most frequently in our cohort, we can see that the allele frequencies in our study were less than 0.001. This is extremely low, which indicates how rare these variants actually are. Anything below 1% or 0.01 is considered pathogenic. So if we also then compared this to other population databases, including NOMAD and H3 Africa, and the 1067G2A variant was not present in these databases, whereas the allele frequency of the 1448CT2 variant was even lower. So if we then take the allele frequency data that we gathered in this study and we extrapolate it to national data, we can then say that per year, less than 5 African newborns and less than 1 Caucasian Indian and mixed ancestry newborn will carry either one of these two variants. So that just gives us an idea of how we can use the genetic information to make future predictions. If we further then have a look at the haplotyping for the specific variants, firstly, the 1067G2A variant, we found a shared haplotype among the patients or the participants in the study that carried the variant. And it had a large minimal overlapping region of 7.2 megabases as indicated by the box. So what this then shows us is that this variant genetically surrounding this variant, there's a lot of overlap between these participants, and that then gives an indication that there is a potential founder effect. So with this analysis, it was also possible to establish that there were no familial relations between these individuals. Since this specific variant has only ever been encountered in the Southern African population, it is reasonable to assume that the founder is potentially in the Southern African Caucasian population. So for the 1448CT2 variant, the same picture was seen. So the minimal overlapping region was smaller. It was a 4 megabase minimal overlapping region. Unfortunately, however, because this variant occurs in other countries and other ethnicities as well, we cannot determine specifically where this founder effect lies. It should also be mentioned that since we do not have a lot of genetic information for the specific population groups that we were investigating, we cannot determine the frequency of this shared haplotype in either one of the cases. But that hopefully will improve as we do more sequencing in South Africa. Right, so if we then have a look at the next assays that we did, the functional assays. To do this, we obtained skin biopsies, and we were able to do this to obtain this from 3 consenting participants and 3 healthy controls. So these included participants 3 and 9 from the variably riboflavin responsive group and participant 5 from the riboflavin responsive group. These skin biopsies were then cultured to obtain primary dermal fibroblasts, and these fibroblasts were genotyped using Sanger sequencing prior to performing any analyses. This was done to ensure that there was no sample mix-up. The first step for the functional assays included evaluating the protein expression, and this was done by SDS page and Western blot analyses. And specifically, this was done for the ETFQO protein to have a look at the structural and functional aspects of the genotype. And then we also did these analyses for the OXPHOS complexes to look at the effect that the potentially less stable ETFQO protein would have on the functioning of the mitochondrial oxidative phosphorylation system. And for both instances, the results were normalized total protein content. All right, so firstly, looking at the findings for the ETFQO protein. So for participant 3, which is shown in red, you can see that there was, this participant showed the largest decrease in stable ETFQO. So only 17% of the protein in comparison to the average of the two control samples, which are shown here as C1 and C2. This was as expected if we consider the pathogenicity of the 287 duplication variant. So as I've mentioned earlier, the 287 duplication variant results in a much shorter protein than the native protein. And for that reason, what we are seeing here most likely is only the protein produced by the allele carrying the 1448C2T variant. So this explains why the ETFQO quantity is so low. So if we look at participant 9, there was also quite a significant decrease. So only 27% of the protein with regards to the controls. Again, as I've mentioned earlier, the 1067 G2A variant is considered to be very pathogenic according to it, or based on the domain in which this variant is found. And it then also makes sense that there would be such a large decrease. However, in combination with the 1448 C2T variant, this does appear to be more than participant 3. So in the case of participant 5, which displayed the 1448 C2T homozygous genotype, a lot more of stable ETFQO was present. This corresponds to previous studies on this variant, but also shows that the specific genotype has an effect on the stability of the protein. If we then have a look at the oxidative phosphorylation complexes, we can see that for participant 3, in figure B, complex 1 was decreased, and in figure E, we can also see that complex 5 was decreased. If we then have a look at participant 9, we see the same results, not decreased as much, but also decreased. So for these two participants, it was hypothesized that if the variant is quite severe, as was the case with these two participants, then the cell will move metabolic pathways away from the oxidative phosphorylation system towards alternative systems that can produce more ATP. If we look at participant 5, we can see an increase in complex 1 with regards to the controls, as well as complex 2. So in this participant's case, or in this specific genotype's case, the hypothesis here was that if the variant or the genotype is less severe, then there will be an increased dependency on the mitochondrial oxidative phosphorylation system, and the OXPHOS complex expression will be increased. This was substantiated by other studies, which found exactly the same thing. And basically explains how the mitochondrion, because the mitochondrion has its own DNA and its own regulatory systems, how the mitochondrion might respond to increase ATP production through the OXPHOS system in cases like this. If we have a look at complex 3, here we specifically analyze the UQ0C2 subunit of complex 3, and an interesting pattern emerged here. So for all of the cases we examined, we saw that there was an increase in this specific subunit of complex 3. Now, recently there was a study published about UQ0C2 specifically, showing that that subunit is the subunit that links ETFQO to the rest of complex 3. And so it was hypothesized that if ETFQO stability is compromised, that the cell might respond by upregulating UQ0C2 in an attempt to better connect ETFQO and complex 3 of the oxidative phosphorylation system. This was further supported by the inverse proportional relationship between the levels of UQ0C2 and ETFQO for participant 3 and 9. So participant 3's ETFQO was the lowest. Here it's higher. And then the same for participant 9. The very high levels that we observed for participant 5 was attributed to this, as well as the upregulation of the mitochondrial complexes, of the OXPHOS complexes. Right, so the final step in my thesis was to further have a look at the flux through the oxidative phosphorylation system. So to do this, we analyzed the fibroblasts using the Ouroboros O2K, and this instrument is a high-resolution respirometer that can be used to determine how efficiently energy is produced by the oxidative phosphorylation system in response to specific substrates that the cells are exposed to. So prior to each assay, this instrument was washed and calibrated, and 2 million cells were prepared in substrate-limited media and added to each chamber of the instrument. The substrates that we evaluated were then added sequentially to the cells following permeabilization of the cells to allow them to enter the cells, and the values, specifically the oxygen consumption rate, was then measured in each case following stabilization of these measurements. So just to explain the different substrates that we used and why we used them. So the first substrate that was added was the fatty acid octonyl carnitine. So the catabolism of this specific fatty acid leads to the production of FADH2, which, as I've mentioned earlier, can then be reoxidized to FAD by the ETF-ETFQO system. So the electrons that come from FADH2, which originally came from this substrate, then feed into the OXPHOS system through the ETF-ETFQO system. The second substrate that we analyzed was pyruvate. So pyruvate is complex 1 specific and leads to the production of NADH. So NADH can then enter the OXPHOS system through complex 1 to produce ATP. The third substrate was succinate. This is a complex 2 specific substrate, which leads to the production of FADH2, and this then enters complex 2 to produce ATP. And the final substrate that we had a look at was glycerophosphate. This was actually just one of the additional substrates that you can add. We didn't expect to find anything with this, but we actually did, at the end of the day, have an interesting result with this one. So this enters the oxidative phosphorylation system at the point of coenzyme Q10, then flows through the system to produce ATP. Right, so if we have a look at the results, firstly, if we look at participant 3's results, we can see that octonyl carnitine, which feeds in through the F pathway, as I've mentioned, was decreased in this participant. This corresponds to the low ETF-QO that we observed. So since ETF-QO stability is compromised, it seems as though ETF-QO's function is also compromised in this specific participant. Then, if we have a look at figure B, at participant 3's response to pyruvate, once again, we see a decrease. This corresponds to the decreased levels of complex 1 subunit that we evaluated. And in this individual, respiration could be rescued by the addition of the complex 2 substrate succinate. If we then look at participant 9, firstly, at figure A, once again, we can see that octonyl carnitine, or respiration through the F pathway, is decreased in comparison to the controls. Again, this corresponds to the decreased levels of ETF-QO, but this is a little bit higher than participant 3. And if you'll remember, the ETF-QO values were exactly the same. An interesting observation here was that in participant 9, respiration through the N pathway, or complex 1, was not decreased. So this was not what we expected to find. And respiration in this individual could be rescued by the addition of pyruvate. Interestingly, if we have a look at the GB pathway, respiration was also rescued by the addition of glycerophosphate in this specific participant. Then if we have a look at participant 5, we can see that the respiration through the F pathway in figure A was comparable to that of the controls. This was similar for complex 1 and for complex 2. So this then again corresponds to the upregulation of the mitochondrial oxidative phosphorylation components as a coping mechanism to cope with the decrease in energy production as a result of the ETF-QO. But it also shows us, if we have a look at figure A, at the normal results that we obtained here, that the compromised ETF-QO in this participant does not affect the functionality of ETF-QO to such an extent. And again, this result has been reported before for this specific genotype. Okay, so this then brings us to the conclusions. So in terms of the clinical and molecular aspects of the study, we can say that in Southern Africa, MADD occurs most prevalently in the Caucasian population. We can say that it is caused by at least 5 pathogenic ETFDH variants. And since we now know what these variants are, this allows us to do rapid screening for them in the future. Of the variants that we identified, there are 2 that are specific and unique to Southern Africa. This is the 1067G2A variant and the duplication 287, the duplication at 287. Based on the prevalence of the 1067G2A variant and the 1448CDT variant, we can also say that those 2 variants will most likely be in a diagnosis of MADD in a Southern African participant. If we extrapolate this to national data, as I've mentioned before, we can then say that fewer than 5 African newborns and fewer than 1 Caucasian Indian or mixed newborns will carry one of these 2 variants, and that these 2 variants are the result of founder effects with that of 1067G2A, likely having arisen in the Caucasian Southern African population. If we have a look at the clinical and molecular results in combination with the metabolic results, MADD can be organized into 3 groups. So the riboflavin unresponsive group, the variably riboflavin responsive group, and the riboflavin responsive group. And interestingly, we can then make assumptions of the disease progression and treatment response based on the specific genotype of the participants. And there is a summary of this available. So if you would like a summary of this, you're welcome to ask. Then in terms of functionality of the ETFQO protein, with the three specific genotypes that we evaluated, ETFQO was the least stable in the 287 1448 compound heterozygous genotype. It was also unstable, but more stable in the 1067 1448 genotype and the homozygous 1448 CTT genotype displayed the most stability. We can also say that from these results that MADD leads to secondary mitochondrial derangement, and it appears that if a participant presents with a very severe genotype, that the dependency on oxidative phosphorylation for the production of ATP will be decreased, whereas a mild genotype will increase dependency on the oxidative phosphorylation system. In terms of complex 3, the UQ-CRC2 subunit seems to increase with a decrease in ETFQO stability, and this appears to be a coping mechanism for the cell to try to restore the connection between ETFQO and complex 3. These results, the oxidative respiration results that we found were also corroborated the structural findings. And from this, we can see that different genotypes can be rescued by different substrates, which leads us to believe that there's potential merit in patient-specific treatment. And with that, I would just like to then acknowledge our various collaborators, my study supervisors, and my colleagues, as well as the funding platforms that made this study possible. I'd again just like to thank the NMP and Pfizer for this opportunity, and of course for you for your attention. Thanks so much.
Thank you, Michelle. Fascinating work. Well done. Okay, so you are welcome to post your questions in the Q&A section. While we are waiting for some questions, Michelle, maybe I can ask you the 1067 G2A variant had a decreased protein concentration. However, that's not what you would expect, right? With just a point mutation. I mean, the protein is non-functional, but the concentration should not be affected unless, you know, there's a complex that doesn't form, and then that influences stability of the protein. So what is your views on that? So the premature truncation you can understand, but
yes,
the point mutation is a little bit more difficult for me to understand why the protein expression would be lower.
So I think here, unfortunately, we weren't able to do specific analyses and docking analyses to see how the specific variant affects the conformation of the protein, and that's something I would really like to follow up on. But the assumption here is that this variant leads to amino acid substitution, which decreases the stability of the protein to such an extent that the mature protein is not produced in the same quantity because it is unstable. It is broken down more easily by cellular processes before it can actually be assembled into a stable functional protein. So while the protein itself is not truncated, it has to do more with the stability of the protein.
Okay, no, I got you there. See, there's still no questions, so maybe I can ask some more questions. The
You're
flux
welcome.
studies that you mentioned earlier on in your presentation. So I listened to previous presentations also from the Dutch where they talk about the utility of the flux studies in MCAT and VLCAT to determine whether a patient will present clinically or not. So do you think there's any place for these flux studies in MADD patients, given that there seems to be fairly good genotype-phenotype correlation, and that you would probably put all of them on riboflavin treatment in any way. So do you think there's a place for those kind of studies in these patients or not?
Right, so the default is, if someone has MADD, is to put them on riboflavin treatment. It is inexpensive and it works very, very well. L-carnitine is a little bit more pricey, but not everybody requires that. With these types of analyses, the flux analyses, they're generally not considered expensive analyses to perform, and the Dutch do still use them in the current laboratories and for diagnostic purposes. So I think it's definitely something to consider. I don't know if it will give you any additional insight to how the patient will progress throughout their disorder, but it will definitely help with the initial diagnosis to pinpoint the specific fatty acid oxidation disorder that the participant has. Oh, sorry, the patient has. The specific flux assays that I was referring to here are the labeled enzyme analyses that you can perform to distinguish VLCAD from MCAD from MADD. So to distinguish between the different fatty acid oxidation disorders. So that's I know what some of the analyses that they developed, I think in the 90s, and that they still actually use. And then something else that's not specifically a flux assay, but something that I know that they do that we don't, is they culture fibroblasts or leukocytes, if I remember correctly, from these participants, and then they do acyl-carnitine analyses on these specific cells, and that also gives them some insight to how the participant, the patient will present. So because you don't have the problem of medication and different diets and different environmental factors, it's a lot more controlled, so that in a research or from a research point of view, gives you a little bit more control to see how the specific genotype presents with regards to another or control participant.
Okay. Strangely enough, there's still no questions, so maybe I can ask one more. What's the time? Yeah, we're just about over our time. Maybe just a last word. So all of your patients were either Caucasian or mixed ancestry, and when you did your calculations there, it basically suggests that we should see about 5 Black patients a year and 1 Caucasian patient. So any final words on, why haven't we seen the Black patients?
We actually have seen them. It's something that, sorry, I just didn't mention, but we have seen MADD in African cases and in Indian cases before. It's not as common, but there are reports of this. For the purpose of my study, unfortunately, I could not locate these participants. They were lost to follow up before the onset of my study. So I would have loved to include them, and we did advertise very widely to see if we can include as many as possible. But unfortunately for these 2 groups, it was not possible to do so. I do think, however, that MADD, or if we have a look at the results that we currently have, with the majority of the cases being in the Caucasian population, and then with a few of them being in the mixed ancestry population, that MADD may have originated from let's say populations or smaller populations that originally came maybe from Dutch populations or German populations, but this might not have originally been an African or an Indian disease. But you definitely see them in other populations. There are reported Indian cases, not specifically in South Africa, but in India, that have MADD. You see this in the Asian populations as well. So for African, I'm not as certain what the prevalence here is, but with regards to Asian populations, there's definitely a chance that we might be missing more patients in our own populations.
Okay, there is a just a question from Roberto Biagio. He's asking if the presentations are being recorded. So I think with that question, I'm going to hand over to Ilse just to answer that and for final closing remarks. And I thank you for your attention. And I thank Michelle. Of course, I thank you as well.
Thank you, Prof.
Ilse, so if you can just explain the channel and so on, I'd appreciate that.
Chris, it's Alna here. So Ilse quickly had to step away. So in terms of recording, our CPD presentations are recorded. It's available if we have consent from the presenters to share the presentations. It will be made available on our YouTube channel, and you can also visit our website where you can find the link to the presentations there.
Okay, thank you very much. With that, then I'm going to conclude this session. Again, Michelle, thank you again for your presentation, very insightful. I also just want to mention that these presentations are sponsored by Pfizer, so we thank them for their sponsorship. They do not influence the content of the presentation in any way, the selection of themes or the presenters that we use. So thank you very much for your attention, and we will see you next time. Bye-bye.